Welcome to microbiology classroom. I want to describe and discuss about the general characters of yeast and molds. General characters of molds and yeasts. General characters of fungi. Fungi are a large, diverse, widespread group of eukaryotic microorganisms. These are a large group of organisms. So far, 1,20,000 species of fungi are described and discovered. In 1999, one landmark research paper published estimated that there are 1.5 million fungi on the earth. And the analysis of environmental DNA samples from a soil community revealed a high rate of new fungal species accumulation at the site, and these data supported an estimate of 3.5 to 5.1 million different fungal species. Number of fungal species yet to be discovered and yet to be reported. There are only 1,20,000 fungal species are so far reported. Fungi are highly specialized, non-chlorophyllous, heterotrophic, spore-bearing, eukaryotic microorganisms. Fungi, these are highly specialized microorganisms. These are non-chlorophyllous, do not contain chlorophyll, and they possess heterotrophic mode of nutrition. One of the important characteristic feature of fungi is presence of spores, spore-bearing eukaryotic microorganisms. Fungus is a Greek term which means soft, fleshy, sponge-like fruiting bodies of mushrooms. Fungus, this is a Greek term which means sponge-like fruiting bodies of mushrooms. Fungi not only include mushrooms, fungi include molds, mushrooms, rusters, smuts, toadstools, polypores and yeasts. They are diverse group of microorganisms, diverse group of heterotrophic, non-chlorophyllous, spore-bearing, eukaryotic microorganisms. They include molds, mushrooms, rusters, smuts, toadstools, polypores and yeasts. Fungi has no chlorophyll, so they cannot synthesize their own carbohydrate. They obtain carbohydrate from outside environment or from the host cells. These are heterotrophic in their mode of nutrition. These are chemo-organoheterotrophs. They derive energy by oxidation of chemicals, and they get the reducing power electrons from the chemicals, organic compounds, and these are they are unable to synthesize complex carbohydrate, complex carbon source. They derive all the three sources from complex carbohydrates: energy, reducing power, and the carbon is obtained in the form of complex carbohydrate. So these are chemo-organoheterotrophs. They either live as parasites or saprophytes. Heterotrophic mode of nutrition is two types. They may grow either as parasites or saprophytes, but some are symbionts. They had symbiotic association with other organisms. Fungi are two types, molds and yeasts. Mold, what is mold, what is yeast? Mold is a fungus that grows in the form of multicellular filaments called hyphae. So mold is a fungus that grows in the form of multicellular filaments called hyphae. Whereas yeast, in contrast to the mold, it is single-celled. So single-celled fungi are called yeast and multicellular filamentous fungi are called molds. Molds are a large and taxonomically diverse group of organisms. Molds are large and diverse number of fungal species where they form hyphae grow and hyphae grow and show branching, and they form the mycelium, and they appear fudgy in appearance, especially when grow on the food. The network of these tubular branching hyphae is called mycelium. So mycelium, they form the mycelium, and the hyphae will break, and when they separate again they form the mycelium. In that way they multiply, and they multiply by means of, they reproduce by means of sporulation also. Each spore germinate and form the hyphae, hyphae grow and form the mycelium. Hyphae are generally transparent, so mycelium appears like very fine fluffy white threads over the surface. Molds grow in diverse habitats. They grow on dead and decaying organic matter in soil and foodstuffs. They grow on different foodstuffs such as pickles, vegetables, fruits, bread, and they grow in different foods, and they sometimes they cause spoilage of the foods, and some are aquatic, and some fungi grow as parasites in plants and animals. Some fungi grow in different animals. Some fungi they grow in plants. They grow on the surface of the plants. They grow inside the plants. Some fungi they enter into an association with other organisms. Such mutualistic symbiotic association is called symbiosis. And this symbiotic association includes lichens and mycorrhiza. Lichens and mycorrhiza, these are associations of fungi with other organisms. Lichen is an association of alga and fungus, or fungus and cyanobacteria, whereas mycorrhiza is association of fungus with plant roots. Lichens are symbiotic association of fungus with alga or cyanobacteria. Lichens consists of fungus and an alga or cyanobacteria. Fungal partner of lichen is called mycobiont, and algal partner of lichen is called phycobiont. Fungal mycelium absorbs water and minerals from substratum and transport it to the algal partner or cyanobacterial partner phycobiont. Alga or cyanobacteria, they use this water and minerals and undergo photosynthesis and synthesizes complex organic compounds. These complex organic compound carbohydrates are shared by both phycobiont and mycobiont, both alga and fungi, or cyanobacteria or fungi, cyanobacteria and fungi. So this association is called the lichen. So about 20% of all fungi and 40% of the ascomycetes, around 13,500 species, are lichen forming fungi. So lichens are most commonly observed. Their distribution is very rich in deserts. Deserts are rich in lichens. Mycorrhiza is another symbiotic association of fruits of plants and fungi. Fungal hyphae, they grow on the surface of the roots or within the roots, and these fungal hyphae functions like the root hairs, and they absorb the water and minerals from the soil and transport it to the plant, and plant provides nutrients to this fungi. So this association is symbiotic association. It is called mycorrhiza. The association of roots of plant and fungi is called mycorrhiza. Mycorrhizas are highly evolved. These are mutualistic association between the soil, fungi and plant roots. Mycorrhizas are of different types. The mycorrhiza association or fungi in mycorrhiza include they are the members of zygomycetes or ascomycetes or basidiomycetes, but not the oomycetes. So they belonging to different classes. The fungal partners in mycorrhiza belongs to zygomycetes, ascomycetes and basidiomycetes, but not oomycetes. And the different types of mycorrhizae include vesicular arbuscular mycorrhiza, VAM, is more important mycorrhiza in which zygomycetes fungi produce arbuscules and vesicles, So this type of mycorrhiza show arbuscules and vesicles within the root cortex. So such type of mycorrhiza is called vesicular arbuscular mycorrhiza. This is one important type of mycorrhiza seen in the plants. Ectomycorrhiza, it is second type. Ectomycorrhiza, the name itself indicating the hyphae form a mantle around the roots and hartig net between the root cells. So such mycorrhiza is called ectomycorrhiza, where basidiomycetes members, basidiomycetes fungi, they form the ectomycorrhiza. And there is other type of mycorrhiza, archid mycorrhizas, where fungi produce coils of hyphae within roots of archidaceous plants. Archid mycorrhiza is other type of mycorrhiza. Ericoid mycorrhizas involving the hyphal coils in outer cells of the narrow hair roots of the plants in Ericalis plants. So this is called ericoid mycorrhiza. There are different types of mycorrhiza: ectomycorrhiza, vesicular arbuscular mycorrhiza, archid mycorrhizas, and ericoid mycorrhizas. These are different types of mycorrhiza. The body of the fungus is called thallus. The body of the fungus or mold is generally called as thallus by botanists. The body is called thallus. The thallus of mold consists of branched intertwined filaments called hyphae. The body of the mold consists of a branched intertwined filaments, thread-like structures called hyphae. The whole massive network of these tubular branching hyphae is called mycelium. The molds have three morphological types of hyphae or mycelia. The septate cenocytic hyphae, non-septate cenocytic hyphae, first type septate hyphae. So there are three different types of hyphae. Non-septate cenocytic hyphae, first type. Septate hyphae with uninucleate cells, second type. Septate hyphae with multinucleate cells, third type. So these are three different types of hyphae or mycelia. Non-septate cenocytic hyphae, characteristic feature of lower fungi, and septate hyphae with uninucleate cells and septate hyphae with multinucleate cells. So non-septate cenocytic hyphae, in these hyphae septae are absent, cross walls are absent. Hyphae are multinucleate and aseptate. Aseptate multinucleate hyphae or mycelium is called cenocytic. Second type of hyphae or mycelium seen in higher fungi is septate hyphae with uninucleate cells. The hyphae are septa or cross walls at regular intervals. These septa divide the hyphae into many cells or compartments. Each cell has one nucleus and cytoplasm. The septa may be complete membrane, allowing only limited exchange of ions and nutrients. They may contain central pores. The pore of septum allows movement of cytoplasm only from one cell to other cell, but not the nucleus or nuclei. Septate hyphae with multinucleate cells. These hyphae have septa, but each cell has more than one nuclei. So there are three different types of hyphae are seen in fungi: non-septate cenocytic hyphae, septate hyphae with uninucleate cells, septate hyphae with multinucleate cells. Septate hyphae, septate mycelium is characteristic feature of higher fungi: Ascomycotina, Basidiomycotina, Deuteromycotina. The aseptate cenocytic mycelium is characteristic feature of lower fungi: Mastigomycotina and Zygomycotina. The septum is simple septum in Ascomycotina. In septate mycelium, septate hyphae, if you see, the septa is different types. The septum is simple septum in Ascomycotina, in which the septal pore is very simple, and this septum is called simple septum, which is characteristic feature of Ascomycotina. complex septum is present in Basidiomycotina. Complex septum is seen in hyphae or mycelium of Basidiomycotina, in which the septal pore is complex. The central pore is surrounded by swollen rim like a structure. On either side is capped by a thickened porous paranthosome. This is called Dolichopore septa. Dolichopore septa, the region around the pore is swollen, and swollen rim on either side is capped by a thickened porous septal pore cap called paranthosome. Paranthosome prevents the migration of nuclei from one cell to other cell and allows only the passage of cytoplasm. Dolichopore septa is characteristic feature of Basidiomycetes. The fungal cell contains cell wall. The fungal cell externally contains the cell wall. It is externally surrounded by a cell wall. Cell wall is composed of microfibrils of cellulose and chitin embedded in amorphous polysaccharides, glucan and mannans and protein. Chitin is a polymer of N-acetyl glucosamine, whereas the cellulose is a polymer of D-glucose. So, fungal cell wall, it is composed of microfibrils of cellulose and microfibrils of chitin embedded in amorphous polysaccharides, glucans and mannans and proteins. Chitin is a polymer of N-acetyl glucosamine and cellulose is a polymer of D-glucose. Below the cell wall, cell membrane is present. Cell membrane is lipoprotein membrane and cell membrane is selectively permeable membrane. So, below the cell membrane, cytoplasm is present. Cytoplasm contains membrane bound cytoplasmic organelles such as endoplasmic reticulum, Golgi bodies, mitochondria, microtubules, sap-filled vacuoles, 80S ribosomes characteristic of eukaryotic microorganisms, and non-living inclusions such as lipids, glycogen, oils, valute granules, etc. are present. Between the cell wall and cell membrane, membranous tubules and vesicles formed by invagination of cell membrane are present. These are called lomasomes. The cytoplasm contains one or more nuclei. Cytoplasm contains one or more nuclei. The nucleus is surrounded by distinct nuclear membrane. Central part of the nucleus is rich in RNA, which is called nucleolus. Higher fungi lack motile cells in the life cycle. The flagella are absent in higher fungi. The motile cells, gametes and zoospores of lower fungi except Zygomycotina are furnished with one or two flagella. Flagellum is made up of tubulin. Eukaryotic flagella contains tubulin, whereas prokaryotic flagellum is composed of flagellin. Eukaryotic flagellum, fungal flagellum, is composed of tubulin and it contains 11 fibrils. Forming axoneme, it shows 9+2 arrangement. Two central fibrils are surrounded by 11 doublet fibrils. Two singlet central fibers or fibrils are surrounded by 9 peripheral doublet fibrils. This arrangement is called 9+2 arrangement, which is characteristic of eukaryotic flagella and cilia. Fungal flagella are mainly two types: whiplash flagella or acronematic flagella and tinsel flagella or pantonymatic flagella. Whiplash flagellum has smooth surface while tinsel flagellum is rough and has fine hair-like lateral appendages called flimmer hairs or mastigonemes. Fungi are nutritionally chemoorganoheterotrophs. Fungi are chemoorganoheterotrophs. They derive energy, reducing power, electrons and protons from organic compounds. They derive energy, reducing power means protons and electrons and carbon from organic compounds. So fungi are chemo-organo-heterotrophs. They derive energy reducing power and organic carbon from organic carbon compounds, ready-made organic carbon compounds. They are unable to synthesize complex organic substances themselves. They require ready-made complex organic compounds, prefabricated organic compounds as carbon source and energy source. Fungi grow either as saprophytes on organic matter or parasites on host cells. Most of the fungi, they are saprophytes growing on dead and decaying organic matter. They produce extracellular enzymes and dissolve complex organic matter and absorb soluble nutrients. Such absorptive type of nutrition is found in fungi. Saprophytes absorb the organic nutrients. They absorb these organic nutrients directly through the mycelium from substratum. In some fungi, rhizoids arising from hyphae absorb the nutrients from substratum. You can see this in Rhizopus. Parasitic fungi, they grow as endoparasites or ectoparasites. They grow on the surface of the host as ectoparasites or inside the host cells as endoparasites. The hyphae ramify in intercellular space between the host cells. Such hyphae are called intercellular hyphae. In some fungi, some hyphae penetrate into the host cells. They enter into the host cell. They penetrate into the host cells. These hyphae growing inside the cell are called intracellular hyphae. The intracellular hyphae of some obligate parasites enlarges at its tip. They swell at its tip and form a specialized absorptive organ to absorb the nutrients from host cell. Such a specialized structure present in the tip of the hyphae, intracellular hyphae, is called haustorium. Haustorium, that is the special structure present in the parasitic fungi for absorption of nutrients from the host cells. Reproduction takes place by different methods. Asexual reproduction and sexual reproduction is seen in the fungi. Reproduction by specialized spores formed without sexual reproduction is called asexual reproduction. Reproduction by specialized asexual spores formed without sexual reproduction is called asexual reproduction. Asexual spores are produced by molds. They include sporangiospores, conidia or conidiospores, chlamydospores, arthrospores or idea. These are different asexual spores formed during the reproduction in different fungi. Sporangiospores. What are these sporangiospores? These are asexual spores formed within a sac-like structure called sporangium, formed on sporangiopore. On sporangiopore, a specialized sac-like structure is formed that is called sporangium. In the sporangium, by cleavage of protoplast terminally or laterally on hyphal branches, the cleavage of protoplast takes place. That leads to formation of sporangiospores. So sporangiospores, there are two types. Motile flagellated spores are called juice spores. Motile flagellated sporangiospores are called juice spores, and non-motile sporangiospores are called aplanospores. By bursting of the sporangium, these sporangiospores are liberated out. Each sporangiospore after germination produce hyphae. Hyphae grow and form mycelium. Conidia or conidiospores, these are asexual spores develop either singly or in chains on the vegetative hyphae or hyphal stacks or on branches called conidiopore. On conidiopore they are formed. So on conidiopore they are formed. They are called conidiospores. Conidiospores are seen in penicillium and aspergillus. Sporangiospores are seen in Mucor and Rhizopus, whereas conidiospores are seen in penicillium and aspergillus. Chlamydospores. What are chlamydospores? Chlamydospores are thick-walled resting spores. Chlamydospores are thick-walled resting structures, asexual spores formed in hyphae during unfavorable environmental conditions. Chlamydospores are seen in Coledotrichum and Fusarium. In unfavorable environmental conditions, the hyphae in certain areas become thick, very thick, and thick-walled resting structure is formed. This is known as chlamydospores. The chlamydospores in favorable conditions germinate and produce the hyphae. Hyphae grow and form mycelium. Chlamydospores are characteristic of Coledotrichum and Fusarium. Arthrospores are idea. Arthrospores are produced by fragmentation of hyphae of septate molds. So these are produced by breakage of hyphae, fragmentation of hyphae in septate molds. These are commonly seen in powdery mildews. In fungi, sexual reproduction is also observed. Sexual reproduction involves plasmogamy, karyogamy and meiosis occurring in sequence. Plasmogamy is the fusion of two protoplastes of two gametes or fusion of protoplastes of gametongia, resulting in a cell with two haploid nuclei. Plasmogamy is followed by karyogamy. Karyogamy is the fusion of two haploid nuclei to form a diploid nucleus. The diploid nucleus divides by meiosis. The diploid zygote nucleus divides by meiosis to form haploid sexual spores. In Masticomycotina and Zygomycotina, plasmogamy is immediately followed by karyogamy. The diploid zygotic nucleus undergo meiosis to develop sexual spores. In Oomycetes, vegetative mycelium is diploid and meiosis occurs in gametongia. In Ascomycotina, plasmogamy is not immediately followed by karyogamy, but a brief period of dikaryotic phase occurs and then karyogamy is followed by meiosis. In Basidiomycotina, the plasmogamy occurs between two compatible primary mycelia, resulting in dicaryotization. Dikaryotic mycelium is formed. Fruiting body forms at the tip of the two nuclei fuse and meiosis occur to form four haploid basidiospores. The various methods of sexual reproduction in fungi are gametic copulation, gametongial contact, gametongial copulation, somatogamy and spermatization. These are the various methods of sexual reproduction. First, gametic copulation. What is gametic copulation? Here gametes will fuse. This is called gametic copulation. Fusion of gametes is called gametic copulation. It takes place by three methods: isogamy, fusion of similar gametes; anisogamy, fusion of some dissimilar gametes, anisogametes; oogamy, fusion of ovum with male gamete. So the gametic copulation, it occurs in chytridiomycetes and hypochytridiomycetes. Second type of sexual reproduction is gametongial contact. The two gametongia come in contact but do not fuse. So the male and female gametongia or plus and minus gametongia, they come in contact but they do not fuse. Nucleus from male gametongia migrate through a pore or fertilization tube into the oogonium. So the male nucleus from antheridium migrate through a pore or fertilization tube into oogonium, female gametongium. After fusion, the zygospore develop after fusion, and such type of gametongial contact that is seen in Pythium and Saprolegnia. Gametongial copulation. The two gametongia or their protoplastes fuse and gives rise to zygote that develop into a zygospore. So gametongial copulation is seen in Rhizopus. Somatogamy. Two somatic cells of two different strains of hyphae fuse, bringing the nuclei together. This is called somatogamy. Somatogamy is seen in Basidiomycetes. Spermatization. In some higher fungi, spermatization is seen in Rustus Higher fungi, spermatization is seen. A special male gamete called spermatium is produced from spermatogonium. Female gametangium or ascogonium is formed with a receptive. Spermatia deposit on receptive trichogyne. Male nucleus enter and fuse with ascogonium. This process is called spermatization. It is seen in higher fungi. After sexual reproduction, spores are formed. These are called sexual spores. Sexual spores are formed after the fusion of two nuclei. Oospores are sexual spores formed after gametangial contact in oocysts. Zygospores are thick-walled resting spores produced by gametangial copulation and fusion in Zygomycotina, Rhizopus and Mucor. In Ascomycotina, ascospores are produced in a special sac-like structure called ascus. These are usually 8 ascospores are produced in an ascus. In Basidiomycotina, basidiospores, these are single-celled sexual spores borne on club-shaped structure basidium. In fungi, spores are produced during the asexual reproduction or in sexual reproduction. So these fungal spores produced by both asexual and sexual reproduction are surrounded by protective tissues together, forming the structures called fructification or fruiting bodies. The asexual fruiting bodies include a cerbulus, commonly seen in Colitotrichum, and pycnidium. Example, Phoma. In Phoma, pycnidium is seen. So the sexual fruiting bodies are Cleistothecium, Perithecium, Apothecium, formed in Ascomycotina. Fungi are responsible for causing different diseases in plants. Late blight of potato caused by Phytophthora infestans. Early blight of potato caused by Alternaria solani. Blast disease of rice caused by Pericularia oryzae. Leaf spot of groundnut caused by Cercospora personata and Cercospora arachidicola. Red rot of sugarcane caused by Colitotrichum falcatum. Damping off of tobacco caused by Pythium dibarianum. Loose smut of wheat caused by Ustilago triticii. Covered smut of barley caused by Ustilago hardy. Black rust of wheat caused by Puccinia graminis triticii. These are few important plant diseases caused by the molds. Some important fungal diseases caused by molds. Ringworm of skin or nails, ringworm disease. This is caused by tinea or ringworm of skin, ringworm of nails. Caused by Trichophyton microsporum epidermophyton species. Trichophyton, microsporum, epidermophyton. These are dermatophytes infecting skin, nails, hair. They cause ringworm in humans. Blastomycosis. This is caused by Blastomyces dermatitis, and Coccidiomycosis caused by Coccidioides brasiliensis. Histoplasmosis caused by Histoplasma capsulatum. Bronchopulmonary aspergillosis caused by Aspergillus, or some human diseases caused by molds. Fungi are also useful. Certain fungi are used in ripening of cheese. Penicillium roqueforti and Penicillium camemberti are used in ripening of cheese. Aspergillus oryzae and Aspergillus niger are used industrially to produce amylases and proteases. Aspergillus niger is used for commercial production of citric acid. Mushrooms, these are used as food. Mushrooms are used as food. Agaricus bispora, Olvariella olvacea, Cortinellus berkelianus, pleurotus. These are important mushrooms which are used as food. Yeasts, general characters of yeast. Yeasts are unicellular fungi. Unicellular fungi are called yeasts. Yeasts are unicellular ascomycetes reproducing asexually by budding or fission. Yeasts are unicellular ascomycetes reproducing asexually by budding or fission. Yeasts are commonly egg-shaped and some are spherical, some are lemon-shaped, some are pear-shaped, cylindrical or even elongated. Some yeast form false mycelium called pseudomycelium. Yeast after budding, they are not separated, forming the filaments called pseudomycelium, false mycelium. Yeasts are larger than bacteria. Their size is 5 to 30 microns and 1 to 5 microns. 5 to 30 microns in the length, 1 to 5 microns in the breadth. Yeasts contain cell wall. Cell wall is composed of chitin and glucan and mannose. Glucan is polymer of glucose and mannose is polymer of mannose. Yeast contain cell wall composed of chitin, glucan and mannose. Cytoplasm contains membrane-bound nucleus and membrane-bound cytoplasmic organelles. Below the cell wall, cell membrane is present. Below the cell membrane, cytoplasm is present. Cytoplasm contains membrane-bound nucleus and membrane-bound cytoplasmic organelles. They contain 80S type of ribosomes, characteristic feature of eukaryotes. Asexual reproduction occurs by budding. Budding is the most common asexual reproduction found in yeast. A small protuberance develop on the cell. It forms into a small bud on the parent cell. The parent nucleus divide by mitosis. It splits into two nuclei, and this daughter nuclei moves into the bud. One nucleus moves into the bud and one remains in the cell, and which the bud will continues to grow until it splits from the parent cell. Sometimes bud is attached to the mother cell and gives rise to daughter buds in chain. The chain of cells looks like a mycelium and it is called a pseudomycelium. It is seen in Candida. Pseudomycelium is seen in Candida yeast. So when daughter cell is separated from mother cell after budding, a scar is formed on mother cell. During the life cycle, a mature yeast may produce 24 daughter cells by budding. Asexual reproduction also occurs by binary fission. Example, Schizosaccharomyces. Asexual reproduction occurs by binary fission. Yeast cell first swells and elongates, then nucleus divides, and then two daughter cells are formed by cell division. Sexual reproduction occurs by copulation of two somatic yeast cells. So, sexual reproduction occurs in yeast cells by copulation of two cells. Zygote nucleus divides by meiosis to form 4 haploid nuclei. They again divide by mitosis to form 8 nuclei. Then 8 haploid ascospores are formed. Ascospores they release out from ascus. Ascospore germinate to form vegetative yeast cell. Saccharomyces is a very important yeast. Saccharomyces cerevisiae is commonly called brewers and bakers yeast. It is employed for production of wine, ale, ethyl alcohol, glycerol, invertase, bread, beer and other alcoholic beverages. It is used for commercial production of different beverages. Saccharomyces cerevisiae is used for production of beer, wine, ale, porter, gin, all beverages. They are produced by using Saccharomyces cerevisiae. Ethyl alcohol, industrial alcohol, it is produced by employing Saccharomyces cerevisiae. Glycerol, invertase, bread is produced by using the Saccharomyces. Zygosaccharomyces, Saccharomyces rauxi, Saccharomyces melis, they have ability to grow in high sugar concentrations, and these are osmophilic yeasts. Candida is a yeast producing pseudomycelium and sometimes true mycelium and produce blastospores and chlamydospores. Candida utilis and Candida tropicalis are used as fodder to cattle. Candida albicans is a pathogenic yeast causing oral thrush and cutaneous candidiasis, bronchocandidiasis, pulmonary candidiasis and vulvovaginal candidiasis in humans. Classification of fungi. Ainsworth 1973 classified kingdom fungi into two divisions, Myxomycota and Eumycota. The fungi locking the rigid cell wall are called in the fungi locking rigid cell wall are placed in myxomycota, and fungi with rigid cell wall are placed in eumycota. The fungi into two divisions, myxomycota and eumycota. Myxomycota division. So myxomycota, the body is called plasmodium or pseudoplasmodium. The body of the fungus is called. So the myxomycota is divided into four classes: acrasiomycetes, hydromyxomycetes, myxomycetes, plasmodiophoromycetes. Division eumycota, where the plasmodium or pseudoplasmodium is absent. Body is typically filamentous. So eumycota is divided into different classes. Division eumycota is divided into the subdivisions first, then into classes. These subdivisions: mastigomycotina, zygomycotina, ascomycotina, basidiomycotina and deuteromycotina. So the mastigomycotina subdivision, mastigomycotina, it produces juice spores. It is characterized by production of juice spores. Mastigomycotina is divided into three classes: chytridiomycetes, hypochytridiomycetes, oomycetes. Subdivision zygomycotina is divided into two classes: zygomycetes, trichomycetes. Subdivision ascomycotina is divided into six classes: hemiascomycetes, plectomycetes, pyranomycetes, discomycetes, lobulbenomycetes, loculoascomycetes. Whereas subdivision basidiomycotina is divided into three classes: teleomycetes, hymenomycetes, gastromycetes. Whereas subdivision deuteromycotina is divided into blastomycetes, hyphomycetes, coelomycetes, three classes. However, during the last few decades, significant advances like recognition of the artificial nature of the three kingdoms and five kingdom classification systems, polyphyly of organisms, these phylogenetic systematics, application of molecular techniques in mycology and discovery of new organisms led to making of a phylogenetic classification. Mycologists are gradually accepting the term phylum as a substitute for the category of division, as suggested by most recent International Code of Botanical Nomenclature. Alexopoulos et al. In 1996 classified the fungi into three different groups: kingdom fungi and kingdom Straminophylla and kingdom Protistus. The kingdom fungi contains phylum Chytridiomycota, phylum Zygomycota, phylum Ascomycota and phylum Basidiomycota. Whereas kingdom Straminophylla contain phylum Oomycota, phylum Hypochytriomycota, phylum Labyrinthulomycota. Whereas the protist group contains phylum Plasmodiophoromycota, phylum Dictyosteliomycota, phylum Acrasiomycota, phylum Myxomycota. Thank you for listening the lecture on general characters of fungi, characteristics of molds and yeast.